good morning, good evening, good afternoon, wherever you are. Welcome to another session in this gene therapy course. And today's topic is genetic editing. We are going to use text editors to edit DNA. Okay? And today what we are going to see is how to use these text editors to edit this DNA. This is a plasmid, okay? And this plasmid has information for doing things inside the cells. You see here this fragment. Today we are going to learn how to delete this fragment. You see here, boop, and it goes here, okay? You see here the plasmid now doesn't have the fragment. We have eliminated it, deleted it. And afterwards, what we're going to do is introducing the genes of interest, the genes that we want to express in the cells. You see? It will be very easy. Deleting is just having the sequence in the text editor, pressing the delete, well, highlighting the region that you want to delete, pressing the delete button, then we will have this sequence, all the sequence without this fragment. And afterwards, what we're going to do is introducing a sequence, which is just copy pasting. Okay, we have the sequence for the genes of interest that we want to express. In this case, OSK. Remember, OSK are the genes that will let us to revert the DNA age. So we will revert the age of the DNA using these genes, okay? So we will have the sequence of these genes and we will just copy paste here this sequence. I'm going to show you how, okay? Let's go for it. Let's go to see this. Let's go to see the genetic editing, which is just having the sequence, deleting the region that we don't want, and afterwards copy pasting the DNA sequence that we want. Okay, and we're going to use text editors, a very easy text editor that all of us know, that is Microsoft Word. Let's go for it. Let's remember this plasmid that we are using. Okay, this plasmid has information for this plasmid has two regions. Okay, this first region and this second region. The first region is necessary for amplifying or copying many times the whole plasmid inside bacteria cells, E. coli. Okay, so this region will let us to amplify the plasmid, to have many copies of the plasmid. As you can see here, we introduce the plasmid inside E. coli, and E. coli will recognize the ORI sequence. The ORI sequence is necessary for making many copies of the plasmid inside bacteria. The bacteria recognize the ORI and start copying. And then we have another two pieces of information that are the promoter, a promoter is necessary to express the resistance gene, and the resistance gene has information necessary to survive to the presence of certain antibiotics. So by having all of these, what we do first inside bacteria is copy many times the whole plasmid thanks to the ORI. And then the resistant gene, what we'll do is ensuring that only the bacteria carrying the plasmid will grow, will survive. So at the end of the process, we will have many copies of the DNA. And this second region, we're going to see it now, okay? This second region has a cloning site 1 and a cloning site 2. And in between, it has random information. So we buy the plasmid, okay, from different providers, and it has random information here that we don't need. So what we do is cutting from here, plop, and from here. You see here, and at the end of the process, what we have is the same plasmid without this random region. And why do we do it? Because we want to insert information that we want. And in this case, the information that we want is the OSK genes, okay? So we have the OSK genes sequence in a text editor or wherever. You can get it from PubMed or another resources, another scientific resources. So since we have the sequence of the OS genes and we have the sequence of the plasmid, we just have to insert the sequence of the OS genes inside. That is just copy pasting here. Okay, so at the end of the process, we will have the plasmid with this information for the plasmid amplification, and also we'll have the genes that we want inside. Okay, so the first thing that we need is the plasmid, okay, the backbone that has this information for amplifying the plasmid inside bacteria. For example, for PAAV plasmids, that is, these are plasmids that work very well with genetic material from adeno-associated virus. Yeah, this provider has very nice backbones. So this website has a lot of plasmids that you can use as a backbone, okay, as a template, kind of a template. This website is, you can find it in www.atgene.org/viral vectors/AAV. You will, by typing this, you will get to this website, and in here you can see many backbones, but majority of them are exactly the same. This region that you see here is exactly the same for majority of them, so you can use any of the plasmids that you will find here, because what the people are doing is exactly the same. They use this backbone, they introduce the genes, so you will find that here a lot of different backbones with different genes, so you can use any of them. Okay, so let's go to see how to get the backbone sequence in this website. Let's go for it. Very easy, just type www.atgene.org/viral vectors/AAV. You will get here, so just press one of the options and you will get to this window. Okay, and you will see a little bit of information of the plasmid, what they have done with the backbone, and you will see here a graphic view of the plasmid. Okay, so in here, if you want to see the sequence, you press here. In here you see view all sequences, right? So you press here, view all sequences, and you will get to this window. You will see the full sequence from AdGene. You see here again the graphic view, and here you have the sequence. So if you want the sequence, the only thing that you have to do is copy pasting all this sequence. You select the sequence and copy paste to a text editor, as we have done in here. You see here, this is Word. So the only thing that we have done is copy pasting here from here to here, and this is a view that I have generated. Okay, I have done it in PowerPoint so that you can see it better. You see here the promoter of the gene. The gene is the resistant gene. Okay, so you have the promoter and the gene, and then you have the ORI, and then you have this random sequence. So let's see it. For example, the random sequence is here. This is the sequence. We have I have highlighted it in blue. It goes clockwise. Okay, so we have first the cloning site here. You see here in gray, I have highlighted the cloning site 1 in gray. You see here the random sequence, I have highlighted it in blue. Then we have the cloning site 2 that is highlighted here again in gray. Then we have a more random information in here that is represented by black. Then we have, sorry, by black. Then we have the ORI here. Then we have more random sequences, and then we have the resistant genes and the promoter. You see here, this is the resistant gene and this is the promoter. So it's a very nice habit to copy paste your sequence and to identify by colors the different regions. How do you identify the different regions? Yes, you hover your mouse over the different parts, they will be highlighted here. So the only thing that you have to do is identify in the different regions and highlight it in your Word. So this is a simple way to have in Word well organized your sequence. I have to say that there are many other text editors, for example, text editors that are specific for DNA, and this will be much easier. In Word, it's very easy, and it's even easier using these DNA text editors. Okay, so that is the only thing that we have done is looking for the plasmid, getting the sequence, and highlighting the different parts. So now we have the backbone here and the sequence and everything highlighted. So let's go for the next step. Okay, so remember what we are going to do. We have the backbone with the random sequence, and what we are going to do is just delete in this region this random information, as you can see here, by pressing the delete button. And afterwards, we are going to copy paste the genes of interest. In this case, the OS genes inside here. Okay, so this is the first step. You see here the promoter, the resistant genes, the ORI, then we have the restriction site 2, then we have the random information, then we have the restriction site 1. Okay, see here you can see clearly the award my Word file. You see here restriction site 1, random information, restriction site 2. The only thing that I'm going to do is just selecting this sequence, okay, and deleting it. We are going to delete, to press the delete button. This is the only thing that we have to do, and it's important to respect the end of the restriction site. Okay, it's important to keep the sequences aside, to keep the sequences intact. So we are going to cut from here to here. We are going to delete this region. That's the only thing that we are going to do. Okay, let's see how it will look like. Yeah, we are in this second step in which we have deleted this random region, okay? Because we don't want it. We want something else, the genes of interest. So okay, we have just pressed this. The only thing that I want to tell you here is that I have deleted these regions. You see here, this represents the end of the cloning site 1. You see here, this is the end of the cloning site 1, and this is the beginning of the cloning site 2. Okay, and we have deleted what was in between. You remember this blue sequence? So the only thing that we have done is just deleting, pressing the delete button, and that's all. We haven't done anything else. So let's go for the next slide that is going to represent this third step. Okay, let's go for it. Okay, so in the third step, the only thing that we have done is copy pasting the sequence of the gene of interest. We have the sequence of the gene of interest in another text file here, and we just copy paste in between the two cloning sites, the cloning site 1 and the cloning site 2, as you can see here. Okay, so here we have, remember, we have here in gray the sequence of the cloning site 1, and now you see all these in orange. This is what we have copy paste. So the only thing that we have done is pasting the sequence of the gene of interest in between the cloning site 1 and 2 sequences. Okay, so we have pressed here, boom, and set the right mouse button and select the paste option, and we paste this here, and the result of all of this will be this plasmid with this nice plasmid, that how now it has information for this first part has information for amplifying the plasmid inside bacteria, and the other side has another information that I'm going to explain to you now. Okay, but you already know if you've seen the first videos. So that's all, my friends. The only thing that you have to do is downloading the sequence or copy pasting the sequence, deleting the region that is random, and copy pasting the sequence that you want in between the cloning site 1 and the cloning site 2. That's all, my friends. Let's go for the next slide. Okay, now we have the whole sequence, the sequence that we want, okay, with this first part that is highlighted in blue and this second part that is highlighted in orange. Let's understand what every single part is doing. Okay, let's remember, I have told you, in the first part we have the ORI, which is the sequence necessary for making many copies of the whole plasmid, okay? So the ORIs recognize the bacteria and start producing many copies. And the resistant genes and the promoter, what they are doing is expressing resistance to certain antibiotic. So with this resistant gene, we ensure that only the bacteria carrying these plasmids grow, okay, in the presence of antibiotic in the media. Okay, let's see this second part that we have inserted, that we have copy paste in between the multicloning site 1, multicloning site 2, okay? You see, this is the gene of interest, and the gene of interest is in between these two sequences that are called ITR. ITR. Let's see these sequences before the OS genes, okay? These ITR sequences are necessary to make many copies of the OS genes. So they are responsible for making many copies of what is in between the two ITR sequences. And then also these are necessary for encapsulating all of these inside the adeno-associated virus, as you can see here. So first we have the plasmid. We produce many copies, and then we insert the plasmid inside these cells that are mammalian cells called HEC293, and inside the cells, you see here, the cell will help it to produce many copies of just this, and then the cells will encapsulate these genes inside the adeno-associated virus. After the process, you will have adeno-associated virus with the OS genes. This is the therapy, okay? So now that you know and understand these two ITR sequences that are kind of a label for these OS genes, let's see what these OS genes are doing. Yeah, so this region has information for producing many copies of the OS genes, and also to include the copies inside the adeno-associated virus, right? As I have told you. So let's see the OS genes. Okay, what happened with the OS genes? The OS genes carry the information for the OS proteins, and the OS proteins are the proteins that are going to revert the age of your DNA. In other words, the proteins that are going to make you young again, okay, that are going to get you back to your 20s or your 30s, okay, or have the health of a youngster, okay. So okay, we saw that now we have the adeno-associated virus with the OS genes inside. That the adeno-associated virus are administered to the human body or another animal. They get to the cells, they get inside the cells, they go to the nucleus, and the cap site is dissolved, and it releases the OS genes inside of the nucleus. Once the OS genes are inside the nucleus, they will produce a mRNA molecule that will be translated in the OS proteins. So what you have here inside the ITRs is information necessary to produce the OS proteins. Okay, in another video I will show you how we edit these genes, this OS information. Okay, it's very easy. It's as you have seen with the rest of the plasmid. Well, all these sequences, the backbone, the ITRs, the promoters, the resistant gene, the ORI, everything is available in the website 3W novosumer.com, and it's also well set up. So you can find the whole sequence, or you can find fragments of the sequence, so that you can play with the whole sequence or fragments of the sequence. Okay, gene editing is as easy as that. It's just identifying the parts and copy paste wherever you want, and making a little bit of sense of all of it is just it. So I hope you have enjoyed this video, and I hope I see you in the following videos. Let's have a good day. Bye bye, my friends. Bye bye.